Thank you, Mrs. Patricia. Maybe if before that about the basic science, if in here I would like to present about my topic as a clinician with direct implantation of adipose derived neuron progenitor stem cell to treat Parkinson. As we know, introduction about the brain is control of center and the body, and also coordinating our movement and also a minute emotion. As we know, is a mantra of biology. If the brain cell do not regenerate, so need add a new neuron. But in the present and the future, maybe the brain can induce by stem cell needs will induce endogenous stem cell. So Parkinson is itself is loss of membrane neuron that synthesize the neurotransmitter is called with dopamine, and the prevalence is higher, more over than 60. And about the second palsy and also initial symptom, usually often the mistaken because part of the aging process. And mostly, usually, the symptom is movement disorder, usually early in the hand tremor and at rest and under stress, and rigidity and also resistance. So the pathophysiology of the Parkinson itself can by direct pathway and also indirect pathway. With a neurotransmitter of dopamine, as we know, dopamine is coordinate smooth and balanced muscle movement and control function related of mood also. So many drug convert the dopamine precursor be dopamine in the brain, and also many research use fetal midbrain precursor and embryonic stem cell be a neuron progenitor induced. But in this research, we use a neuron progenitor from adipose derived. So stem cell itself, the clinical trial 2014, usually the goal is the most is regeneration and also non-regeneration. But in the principal disease, the first rank is cardiovascular disease and the second rank is neurological disease. So we have in Surabaya, Indonesia, we have a roadmap until 2013 until 2014. Some of the research in vitro laboratory and some of the research in animal trial and some of the research is for clinical trial to find the safety, efficacy and side effect of the stem cell. So we have a networking from 3 institutions, School of Medicine, Stem Cell Research and Development Center is for basic technology and clinical trial in our hospital, in the government hospital. We have a basic concept of the stem cell therapy for 3 legs, maybe. The paracrine effect of cell, the effect of cell itself, and also homing mechanism. With a possible approach of stem cell for cerebral damage, it's also the Parkinson maybe, there is a 5. The first is reimplantation, harvest direct progenitor of the midbrain, and also the second is for reprogramming from embryonic stem cell, and the third is reprogramming also from the blastocyst, and the fourth is reprogramming from the bone marrow, and the last may be gene therapy with the vector. So with the plasticity of the stem cell, we use a multipotent stem cell. Although the multipotent is 1 degree below the pluripotent, but safe beside a pluripotent of stem cell. So the source, we take autologous and allogenic with adult stem cell. We don't use embryonic and xenogenic, because embryonic in our country is high controversy from political and religious, and the second high immune rejection, and the third maybe 10% can be teratoma. So from stem cell, we take from adipose, we isolation from the abdomen adipose, and we culture in the D7 and after that D14, and we continue to D21. Before we use stem cell, so we make a validation of the cell with pluripotency, contamination, and purity. For pluripotency, we use SOC and OC and NaNOC, and for contamination, we use hepatitis, HIV, and also from the fungal and the purity, we use NOC1 and also L-DOPA. There is with immunocytochemistry, staining from L-DOPA marker, so the most frequent of the L-DOPA present expression is in the fortidine culture. And with a flow cytometry with a NOC1, we also get much of the stem cell. So after processing and culturing and become a single cell and validation with L-DOPA and also NOC1, so we implant this direct with intraventricular. We join with neurosurgery. There is our team, so we direct implantation intraventricular use in operation room. And we have 10 patients with Parkinson. We use evaluation after 3 months with rank scale and Bartle index. Patient number 1, 65 years old with improvement. And also number 2 and number 3, but not in the number 6. The patient with number 6, no improvement, and the patient number 9, worsening and did intracerebral hemorrhage. But from 10 patients, we have 8 patient improvement. A conservative treatment for Parkinson, usually we use still medicine, physiotherapy, health diet, and also exercise. So we take the sample of the research is the patient with no option treatment after we given levodopa for many years, but the patient still no improvement. So this patient is candidate of the stem cell treatment. The stem cell work in this disease based on 2 aspects. The first is paracrine effect because cytokine and growth factor improvement, brain milieu increasing endogenous stem cell growth, and the second because a stem cell repressing itself. And worsening in one patient, number 9, it might be with comorbidity and precipitating factor, maybe from the age, and the second, the patient have comorbidity with diabetes mellitus, so the vascular elasticity is not good anymore, so have after operation getting intracerebral hemorrhage. So in our center we have almost 400 cases with stem cell done intervention. The first rank is diabetes mellitus and also CVA, osteoarthritis, brain atrophy, spinal cord injury, cirrhosis hepatis, sepsis, and also decomcordis and coronary heart disease. It's data in 2015. So in diabetes mellitus, we find after 3-month evaluation increasing of the C-peptide and the insulin, and also decreasing of resistance and A1C. Is the same. For 40 patients with diabetes mellitus with adipose stem cell, we find the decreasing of A1C become 6, and blood sugar fasting become 100, and also postprandial, and also C-peptide and insulin increasing. With immunohistochemistry, improvement of the insulin promoter PDX1, comparing between a progenitor pancreatic stem cell and also mesenchymal stem cell. is also about expression of the glucagon is the same. We also in the cardiac done in the decomcortis and coronary heart disease evaluation in the 15 patient. So the case male 60-year-old. So after evaluation can improvement of the ejection fraction after 3 months for 15% become 47%. And also case number 2, I think so, evaluation after 3 months and 6 months, so ejection fraction become 68. So in our center we use work in the collaboration of team with disease competency, and also in the osteoarthritis, and we use also in the sport medicine. We saw the stem cell, we use mostly autologous and some of the allogenic stem cell. And with stem cell, mostly mesenchymal stem cell and some of the NK cell, nerve cell, and hematopoietic stem cell depend on disease variation. I think it's our report in the stem cell in our center in Indonesia. Thank you.
Thank you. Are there questions?
The levodopa, you say it's converted to dopamine. What's the conversion rate?
Excuse me?
Sorry. The levodopa, you said, is converted into dopamine in the patients. Is that in all patients or just some?
All patients.
All of them?
Yes.
Without exception?
No, no exception. Yeah. But the patient is all have levodopa, maybe suffer with Parkinson over the 5 years. So there is no option treatment anymore for them. Yeah. Thank you.
Other questions? Okay. I'd like to present you with the token from the meeting. Certificate of recognition.
Thank you. Thank you very much. You're welcome.